Okay, good morning, everyone. Welcome back. I hope that you are doing well. And hey, think of it this way. Do you realize that basically today you are at the halfway point of the course, right? This today marks 4 weeks into our semester. So starting next Tuesday, you'll be on the downside of our semester. So things are moving along very quickly. So, congratulations for hanging in there. Okay, so if you can hear me okay, please type yes into the chat window. Okay, great. That's great. Thank you so much. Okay, so just a quick reminder, obviously you know that you have your lab exam that must be completed today, by the end of today. I know that a few of you have already done that, and that's fantastic. So for the rest of you, please remember to complete that lab exam by the end of today. Are there any questions or concerns at this time about the lab exam from a technical point of view or anything? Do you have any concerns that you need to express right now? Okay, Julia, you asked how much is it worth? Let me get the syllabus out, and I can tell you specifically. The lab exam, this first lab exam that you are taking is worth 10% of your course grade. 10% of your course grade. Okay, any other questions?
Yeah, just to make sure, the lab, we have until midnight to complete the exam.
You do,
okay.
Emily, yes.
Yeah. Okay.
You know, try not to wait that long, but you do. Okay. Anything else?
Yeah, if I cut in and out here, I'm actually on mobile. I'm on a drive to New York right now, just so you know.
Okay, Matt, thank you for saying something. Okay, so let's begin then. What we're going to do is jump heavily into chapter 7. You know, we did get that started, and I introduced you to the ideas of neurons and glial cells, different kinds of neurons like interneurons. We talked about the myelin sheath and Schwann cells. We talked about the functions of the myelin sheath, and then we went on and talked a little bit about that biological weapon at the end of last class. So before I start new content, do you have any questions so far from anything we've been talking about? Okay, not seeing anything, so I'll assume you're okay. All right, folks, so I'm going to go to the chat, to the Microsoft Word document. Okay, so let's do that. Okay, so the chat window is up. If you need to communicate, please feel free. And we now have the Word document up as well. Okay, so let's start talking about what communication in the nervous system really means. Okay, so if you've read through this chapter, you already have an introductory idea of this. Okay, within the nervous system, there's two major types of communication. Okay, the first type of communication is, excuse me, the first type of communication is of an electrical nature. Okay, so there's an electrical communication that occurs. Okay, this communication is also known as a nerve impulse, which is also called an action potential. Okay, so if I use the phrase nerve impulse or action potential, or I'm talking about the electrical signal that runs down a neuron, I'm talking about the same thing. Okay, now this electrical signal or nerve impulse or action potential, okay, this electrical signal is used to communicate within and along individual neurons. It's not used to communicate, well, it's not used to communicate in between cells that frequently. I won't say it's never used to communicate between cells. It's dangerous because there's always an exception to the rule. But for the most part, how we teach this is that generally speaking, the nerve impulse is a form of a communication that happens within a neuron, but not in between cells, not in between neurons. So in your notes, I'm typing used for communication along and within an individual neuron. Okay. The second form of communication within the nervous system is biochemical. Okay. Biochemical communication involves the release of special substances that I know you've read about and you know something about a little bit. These things are called neurotransmitters. Okay. And And every year, scientific research uncovers new molecules that get categorized as neurotransmitters. Same thing happens for hormones. Research every year discovers new molecules that can be defined as a hormone. So, please don't think, as sophisticated as our knowledge is in 2020, that we have all this stuff figured out, because we certainly do not. Okay. All right, so biochemical involves the release of neurotransmitters, and this is used for communication in between cells, for one cell to communicate to a neighboring cell. Okay. All right. So the cells that are communicating to each other could be one neuron communicating to another neuron, or it could be a neuron communicating to a muscle cell, a muscle fiber, such as you find at a neuromuscular junction, which stimulates the muscle cell to contract. Okay, so this is going to tie in. We're going to be coming back around and tying in with concepts from chapter 6 at the end of our conversation today. Okay. So what I'm going to do at this time is show you a short video. It's like a 4-minute video about communication in the nervous system, and use the video to put into your mind some visualization, some imagery, so that as we talk about these things today, you have a reference point to be thinking about how this would work, okay? So bear with me. I'm going to bring up a YouTube video and show this to you. Okay, the video is Nerve Impulse Mechanism 3D Animation by McGraw-Hill. I will send you a link to this video later today. Okay, so let's get this started. And here we go.
The pain you experience when you burn your hand results so quickly in an action by your muscles. Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane, by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be re-established. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, re-establishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
The nervous.
Okay, folks. So that was, depending on your perspective, if you've not dealt with this concept before, that could have been a fairly scary video because there's some fairly complicated things going on, and we need to talk about this video today. But before I do, and you've only seen it one time, do you have any questions about what was on the video? Okay, I'm not seeing anything, so let's begin our journey together to try to explain the content of this video. Okay, so we're going to take the electrical communication first and talk about that, and then at the end, after that, I'll talk about the biochemical communication. Okay, so let's break this down. Okay. Now, there's a nuance here that I would like you to appreciate, okay? So, your textbook, and basically every textbook I've seen, your textbook tells you that a nerve impulse is the same thing as an action potential, okay? And I think that's fairly true. And if you tell me that on an exam, you would get credit. But I think there's a slight philosophical difference between these two ideas that might be helpful for you to appreciate. Okay? So, let's see here. Okay. I'd like to do something with you here and get your participation or response in the chat window. Okay? So just humor me for a moment, okay? Folks, I want you to think about a choo-choo train in your mind, okay? Get an image of a choo-choo train in your head, in your mind, okay? Go ahead and do that. Think about a choo-choo train, okay? Now, question coming through the chat window. In your vision of a choo-choo train, is your train made of a single car or multiple cars linked together? Okay, just give me your honest answer in that. In your vision of a choo-choo train, is your train made of a single car or multiple cars? Okay, take a look at the chat window, folks. Okay, you can see that we have a pretty even distribution of single and multiple. And I guess if I count them up, right, we got 1, 2, 3, 4, 5 people that answered multiple, maybe 6 people that answered multiple, and another close. It's almost 50-50. It's almost 50-50. Okay. So my point is that at least some people think of a choo-choo train as a single car. And other people, myself included, honestly, every time I envision a choo-choo train, I think of a long train that's made of multiple cars. So the philosophical distinction is this, and it's a philosophical question. It does not necessarily have a right or wrong answer. But for those people that thought of a choo-choo train as a multiple car object, can you imagine a choo-choo train as a single car? I mean, is a choo-choo train a train if it's only made of one car? That's a philosophical question, okay? And so you can debate and argue it as much as you want, but here's my point, okay? And I'm typing this into your notes, okay? Consider a nerve impulse as a multi-car choo-choo train. Okay, that's how you should think of it. You should think as a nerve impulse as a multi-car choo-choo train. And you should consider an action potential as an individual car of the multi-car choo-choo train. Okay, so again, we could step back and ask ourselves, well, is an individual car of a multi-car choo-choo train the same thing as the train itself? No, that's a philosophical debate that we don't need to have right now, but it's something to think about. Is there a distinction that should be made between a multi-car train and an individual car of the train? And I would argue for our concept of physiology right now, you should be aware of this distinction. Think of the nerve impulse as an electrical signal, which is analogous to a multi-car choo-choo train. And consider an action potential as an individual car, a single car of the multi-car choo-choo train. Folks, are you with me right now? Is this making sense? You know, the old idea is that if you're educated, you should be able to hold two opposing ideas in your head at the same time and not lose your sanity. Okay, so these are sort of splitting points where we're splitting single hairs here, but philosophically, I think it's worth thinking about this. Okay, any problems, any questions? All right, so let's talk about what an action potential is from a physiology perspective, okay? An action potential is an electrical event, okay? It's an electrical event. Now, it might be difficult to know what I mean by electrical event, okay? Think about, you know, an event is simply a moment in time. Like your birthday is an event. July 4th was an event. It was a holiday. It was an event. Okay, Christmas is an event. Your wedding is an event. Okay, it's simply a moment in time. An action potential is an electrical event. It's a moment where something with electricity happens. Okay, and so an action potential is an electrical event that is made of two substages. Okay, those two substages. Can anybody tell me what the two substages of an action potential are? I'm going to type that into the chat window. What are the two substages of an action potential? Anybody know? Okay, Alyssa, you've answered something there where sodium ions enter the neuron. Alyssa, you're not wrong. I mean, what you're telling me is relevant for our conversation. But what is it called when sodium enters the neuron? There's a name for that. Okay, so Alyssa, you say sodium enters and potassium leaves. That's good information. But there's a name for when sodium enters the neuron. What's that called? Okay. All right. Let's see if I can help you along. Okay. All right. Good. That's what I like. Much better if you dig it out yourself than if I tell you. Yes, Alyssa, depolarization is the answer. Okay. An action potential is an electrical event made of two substages. They are depolarization and repolarization. Okay. Depolarization and repolarization are substages of an action potential. Okay, so think of it this way. An action potential is an electrical event. Well, an analogy would be the event of turning the light switch on your wall on or off, right? You have a light switch on your wall. That light switch could be flipped up to turn the light on. That would be depolarization. That light switch can be flipped down to turn the light off. That would be repolarization. So flipping the light switch up is one substage, flipping the light switch down is the second substage, and both of them are part of the event that you can manipulate the electrical switch on the wall. Okay? All right. So, bear with me a moment. Okay? So, to get any further, gang, I need you to appreciate something here. Okay? So, I'm going to go to a PowerPoint drawing. Okay? All right, so up on your screen now, you should see an empty PowerPoint window here. Okay. All right, so let's take a look at what this stuff really means. Okay. What I'm going to do is draw two segments of a neuron. Okay, and the first segment here and then the second segment over on the right side. Okay. All right, so on the left side, we're going to have a neuron at rest. Okay. And on the right side, we're going to have a neuron that is electrically communicating. So it's electrically active. Okay. Sorry for the rough drawing here. Hard to do on these pads that we get. All right, so the neuron is at rest on the left-hand side. Folks, what I want you to appreciate, okay, so, I mean, you understand that what you're seeing here, right, like this whole blob here could be the axon of a neuron. The axon of a neuron, and you appreciate that, you know, we have the plasma membrane here. The black lines are the plasma membrane. And so we have inside the neuron, okay, you have inside the neuron, and you have outside the neuron. Okay, so this is also outside on top. And this is part of the inside there. Folks, if you can see this okay at your home, please type yes into the chat window. Okay. Okay, thank you. All right. Okay, good, 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 good. Okay. So what I want you to appreciate is that, first off, when you talk about the inside of a typical eukaryotic cell, the inside of the typical eukaryotic cell, where the cytoplasm is, right? Basically the cytoplasm, it's going to be made of a variety of molecules. There's going to be a variety of molecules present. You know, there's going to be dissolved solutes. So let me define a space inside here, which I'm going to define as the interior of the cell. Okay. Please understand that you're going to have a bunch of positive and negatively charged ions and molecules that are in this space. Okay. But if we were to count them up, we would get roughly 50% positive ions and molecules and 50% negative ions and molecules. And what I'm trying to tell you is that the majority of the interior of a cell is electrically neutral. Okay, most of the interior of the cell is electrically neutral. Most of the interior of a cell is electrically neutral. Hope you can draw this for yourself at home, because by the time you get my horrible drawing sent to you, you may not understand what it is. Okay, so please try to draw and follow along. Now remember, the interior is electrically neutral, not because there are no charged materials. There's a whole bunch of them. It's just that we have a roughly equal balance of positively charged substances and negatively charged substances. Positively charged molecules and ions, positive and negatively charged molecules and ions. So it's roughly equal, so the inside tends to be electrically neutral. Okay, not electrically neutron, electrically neutral. Okay. Now, where things change are at the outside and inside border of the cell, at the plasma membrane, okay? You can see from your own textbook on page 119 that embedded in the plasma membrane of the neurons is a variety of ion channels, okay? Now, to be honest, and as you might anticipate, the variety of ion channels that actually exist embedded in the plasma membrane of a neuron is more involved, more complicated than your textbook is telling you, okay? So don't worry about that, because, you know, we're just getting started here. But suffice it to say that you do have these sodium and potassium channels, okay? So I'm not very good at drawing. I'm just going to put a couple things here in the membrane, and I'll make them blue, okay? I'll make them blue and have different shapes, okay? So there's these ion channels embedded in the plasma membrane of the neuron. And long story short, they work in concert with each other, like a symphony, like an orchestra, okay? An orchestra is made of different instruments, right? Okay, we know if you're in a band, you might have a saxophone, a clarinet, a flute, you have some drums, okay? So there's different instruments, but everything together is the band. So this different group of instruments, these different proteins embedded in the plasma membrane of the neuron, work together. So while the neuron is at rest, the overall charge on the inner surface of that membrane is going to be kept negative. Okay, the inside surface of the plasma membrane is going to be negative. And the outside surface of the plasma membrane is going to be kept positive. Okay, so the drawing's getting very busy at this point, and I don't want to draw too much to make it even worse and confuse you. But suffice it to say that while the inside interior of the cell is electrically neutral, along the plasma membrane of the cell, it is not electrically neutral, because the proteins embedded in the membrane maintain a separation of charge so that when the cell is at rest, the inside is overall negative and the outside is overall positive. Folks, is this making sense? Are you with me?
With it keeping the interior being overall negative and the exterior being overall positive, that keeps the cell electrically neutral in terms of the system, does it not?
It does keep the cell electrically neutral, but it keeps the localized space around the membrane not neutral. In other words, it's a separation of charge. These proteins are designed, it's a little bit more complicated than we're sharing with you at this time, but these proteins are designed basically to move positive ions, sodium and potassium around. And the truth of the matter is that for every 3 positively charged sodium ions that go out, you have 2 positively charged potassium ions that come in. So you have more positive out than you have positive in, which means the inside is a little bit more negative than the outside. Does that make sense?
Yes.
Okay, good. Other questions at this time? Now, we're not going to get into the heavy-duty chemistry of this. That's beyond our course. But you know from reading your book, and I think from seeing the video, and I hope that you watch the video again on your own study time, that maintaining this separation, okay, I'm saying it slowly, separation of charge across the plasma membrane, where there's more sodium outside and less potassium inside, making the inside less negative, making the inside less positive, meaning more negative than the outside. The outside's positive compared to the more negative inside. That requires an expenditure of ATP because the proteins embedded in the channel need ATP to move the ions against their concentration gradients in the opposite direction than Mother Nature thermodynamically would want them to move. Okay. So, questions before I continue? All right, so I'm going to get back to the Word document in just a moment and type some of these things up so they're a little bit prettier for you. But before I leave the PowerPoint presentation here, you have to appreciate that what happens during neuron communication as we go from the left drawing to the right drawing is that when the neuron is electrically communicating, the difference across the plasma membrane, the charge difference across the plasma membrane will momentarily switch, like a light switch being turned on, so that at some moment of time when electrical communication is happening, the inside will become momentarily positive and the outside will become momentarily negative, okay? So if you see that switching of the charge difference across the plasma membrane, you know the neuron is not at rest anymore. It's actively trying to send information from one location to the next. Okay. All right. Questions again? Okay. So bear with me. We're going to be coming back and doing some more PowerPoint drawing here. So hopefully you have some colored pencils nearby. Okay, but let me go back to the Word document. Okay, and in the Word document, we've already stated that we have depolarization and repolarization that are substages of this electrical event that we're describing as an action potential. And remember, an action potential is an individual car of our choo-choo train. Okay, so now depolarization, depolarization is the substage in which the charge across the plasma membrane momentarily switch. So I'll define it this way. Depolarization is the event, the substage in which the inner surface of the plasma membrane becomes momentarily positive, and the outer surface becomes momentarily negative. Okay? So for the inside to become momentarily positive, we have moved away from the resting state of the neuron. Okay, so depolarization is always going to be part of the neuron being electrically excited and trying to communicate to another location. Now, just like your light switch on the wall at your house, right? You're going to flip the light switch up to turn the light on. But presumably there's going to be a moment in time when you don't want that light on anymore, so you're going to turn it off by flipping the switch back down. That's going to be repolarization. Okay, so repolarization is a substage in which the inner surface returns to being negatively charged and the outer surface returns to being positively charged. So typing it all out, the substage in which the inner surface of the plasma membrane returns to its negative state and the outer surface returns to its positive state. Okay? So once repolarization has again flipped the charge back to the resting state, the neuron could stay there relatively indefinitely until some kind of stimulus triggers the neuron to want to communicate again, in which case you'll start a new depolarization event. Okay. All right. Okay, folks. So, any questions before we continue? I want to give you plenty of time to ask questions because this is not an easy concept. Okay. All right. So what I need to do next is explain to you the idea of an action potential and its role as in the bigger choo-choo train of a nerve impulse that happens in the nervous system. And there's two scenarios that we need to talk about. We need to talk about how the nerve impulse takes place in an unmyelinated neuron. And then we also need to explain how it takes place in a myelinated neuron, okay? So to do that, I'm going to have to do some horrible drawing for you again. Okay, so I'm going to put into your Word document, okay, see PowerPoint drawing about electrical communication along unmyelinated and myelinated neurons. Okay, so please draw these along with me. It will help get these ideas in your head. All right, so here we go. All right, so back over to PowerPoint. Going to insert a new blank slide here. Okay, and okay. So let's take the scenario where you have communication along an unmyelinated neuron. Okay, so the most likely area of your body where you're going to find the unmyelinated neurons is going to be in your brain, right? Any place where the distance that the electrical signal needs to transmit is relatively short, more likely you're going to find unmyelinated neurons. Okay, so communication, so we'll say this way, nerve impulse in an unmyelinated neuron. Okay, so I'm going to draw a section of a neuron, and this could be anywhere in any part of the anatomy of a neuron but I think it makes most sense simply to draw it out as an axon, because you know an axon is this long cytoplasmic extension that comes off the cell body and is used to transmit information away from the cell body so that the neuron can communicate eventually to another cell. Okay, so we'll just say once again that this is part of an axon here. And again, similar to the other drawings, we have the inside and the outside. Okay. So we know, okay, this is hard. It's hard to explain, and it's going to be hard for you to appreciate this, because the picture you have in front of you has to literally change from one moment of time to the next moment of time, to the next moment of time. In other words, the image of what's going to happen is going to be different from now to now to now. In other words, there's a sequence of time-dependent events where different things are happening at different sequences of time, okay? So the best we can do is try to color code some of this information. Okay. So what I'm going to do next is I'm going to divide this axon into some sections here. Okay. And I want you to understand up front that these sections I'm drawing in your notes are artificial. They don't really exist in the true biology of the body, but I'm putting these sections in as an artificial way to help you understand a bigger concept. Okay, so I'm drawing a dotted red line here to break up the axon in multiple sections. Again, the multiple sections are artificial. They don't really exist, but they're being used to help you understand something. Okay. So I'll put down here in the right corner that the dashed line is artificial. Okay, so you know that it's not real, but I'm using it to help you understand something. Okay. So you know, when the cell is at rest, we've already told you that at rest, the inside surface of the plasma membrane is going to have what charge? You tell me in the chat window. What charge is going to be on the inner surface? What charge should I put on the inner surface of the plasma membrane? Okay, Jennifer, thank you very much. It's negative, that's right. So we have some negative charges here, okay? And I'm just going to draw them in a little bit here, right? And the outer surface, of course, is positive. All right. So this will continue along the length of the membrane. I'm just not going to draw it all in because it takes time. Okay. So we have a neuron that's unmyelinated and it's at rest. So it has a resting electrical membrane potential where you have more positive ions outside than inside. So the outside's positive, the inside's negative. Now, something's going to happen, okay? That neuron's going to generate an electrical signal, but it will only do so in response to a stimulus. In other words, this neuron's going to begin communicating in response to some kind of stimulus. So up here in the upper left corner, I'm going to simply put the old cartoon bubble, like if you've ever watched the old 50s and 60s Batman cartoon, right? Or read any comic books, okay? There's got to be some form of stimulus, okay? So this opens up a question, okay? What are examples of stimuli that can activate a neuron to leave its resting state? And I'll start, okay? So I have a question in the chat window. What are examples of stimuli that can activate a neuron to leave its resting state? I'm going to give an example, and then I want you to give me some examples. Right now, you have neurons in your inner ear that are responding to the sound of my voice, so you can understand the conversation we're currently having right now. So one example of a stimulus would be sound. Okay, sound waves in the inner ear. That is definitely an example of a stimulus that will activate auditory nerve endings. Can you give me some other examples of stimuli that would activate different types of neurons in your body? Okay, chewing food. Okay, absolutely, Jennifer. So, when you chew food, you're going to release molecules on the surface of your tongue, which is going to activate the nerve endings that are associated with your taste buds so that you can perceive the flavors of the food that you're enjoying. So that was very nice. Matt, light is absolutely a big one, right? You're activating the photoreceptors at the back of the retina in your eyeball, right? So that you can see your computer screen or your book that you're looking at, or you can see a family member in the room with you. Music would again be part of the sound waves. Touch, absolutely, Fiona, right? Last time you had a nice, you got a nice hug from somebody, or you gave somebody a hug, right? The skin of your body is compressed, so that activates nerve endings, so you can feel the gentle pressure of the hug, okay? The sight of my beautiful drawing, that should definitely activate nerve endings. Nicely done. Since feeling heat on the body, the warmth of sunlight, right, that will activate temperature receptors embedded in your skin. All of that stuff, folks, are example of stimuli. Now a lot of the stimuli that we talked about came from outside your body, right? And that's good, because we want our bodies to be able to respond to our environment, right? If there's a thunderstorm overhead and you see a flash of lightning and then you have this giant thunder crack, you probably jump out of your seat. But that's a way for your body to get alerted that, hey, there's something threatening outside, right? So our body should respond to stimuli outside ourselves. And if we're being honest, your body does also respond to stimuli that are internal. Best example I could give you is, when your blood vessels, particularly large arteries of the body, when they stretch out because the heart's pushing blood through them, that will send that stretching of your internal anatomy through blood circulation will also activate nerve endings that will send information to the brain to tell your brain about how well blood circulation is achieving its goal in the body. Okay, so there's actually internal things that are stimuli as well. But I think you have the idea here. There's got to be a stimulus. It will activate this unmyelinated neuron. Okay, so questions right now? Okay, all right, so let's keep going here. So let's assume we've had a stimulus. The stimulus might be, the sight of a sunrise or the odor of a skunk outside your window. Whatever it might be, there's a stimulus. When that happens, okay, I'm going to change the color I'm using to draw with. Okay, when that happens, let's see, I'll go to orange here. When that happens, the ion channels, which are embedded in the plasma membrane of the cell, they will move the ions in opposite directions so that in the next moment of time, the inside becomes positive and the outside becomes negative. Okay? So somebody tell me, at this moment of time, when the inside of this first section of the neuron has become positive, what substage of the action potential is occurring? Okay, the stimulus is causing the electrical charge across the plasma membrane of the neuron to change. Okay, the stimulus is changing the charge across the plasma membrane. So what is it called when the inside becomes momentarily positive? It's called depolarization, Quinn. It's called depolarization. So, okay, the red here, or excuse me, the orange, sorry about that. Okay, so down here, I'm trying to give you some notes. Okay, so I'm trying to give you some notes down here. Okay, so what's happening here in this first section is we have depolarization occurring. Okay, depolarization is occurring. All right. So, let me momentarily stop sharing because I want to throw an idea at you. I want to see you to do this, okay? All right. So, if you are able to turn on your video camera, please do that, okay? How many of you have been to a football game or a sporting event of any kind where you participated in a wave? You know what I'm talking about? The wave at a football game where you have people in the bleachers stand up and they sit down. You know what I'm talking about? Please raise your hand if you know what I'm talking about. Okay. All right. So gang, let's think about the wave at a football game. Isn't it true for the wave to make any sense that what has to happen is one group of bleachers stands up, and then what does the second group of bleachers do? What do they do? What does the second group of bleachers do? So Pete, you say they're getting ready. Okay, so the first group of bleachers has stood up. Now what should the second group of bleachers do? Okay, Quinn, you said stay down. That's why this is difficult to communicate, because for the wave at a football game to look normal, you have the first group of bleachers stand up at one moment of time. In the next moment of time, what does the second group of bleachers do? Don't they stand up? And then what does the first group of bleachers do? They sit down, don't they? Folks, I need feedback from you. Am I getting across to you? Okay? Okay. Okay. So I will be the first group of bleachers, and then I want you all to be the second group of bleachers. Okay? I've stood up. What should you do? You stand up, so I sit down. Okay, good. That's what I need from you. So go ahead, you can put your arms down, but at the football game, you have one group of bleachers stand up, second group of bleachers stands up, first group sits down. Third group of bleachers stands up so the second group can sit down. Fourth group of bleachers stands up so the third group can sit down. You get the visualization I'm trying to give you at this time. Okay? So think of those different sections of bleachers as the different artificial sections in the axon that we drew on the PowerPoint drawing. Okay, I'm going to go back to that. Okay? So here we have the different sections of bleachers, right? Here's section 1 of our bleachers. Right, here's section 2 of our bleachers. Here's section 3 of our bleachers. Okay? So what's going to happen is this, gang. When the first section of bleachers has depolarized, what's going to happen is the next moment of time, right? This is a time-dependent thing. So at the first moment of time, the first section depolarizes. At the next moment of time, the second section will depolarize. Depolarization. The reason that happens is because the first section, these positive ions, they literally move over into the second section and change the shape of the ion channels in the plasma membrane so that positive charge comes in and negative charge goes out. Okay? All right. So hold on a second here. Question in the chat window. Question in the chat window. After the second section has depolarized, what should the first section do? Stay with your wave analogy at the football game. After the second section has depolarized, what should, okay, it should repolarize. It should repolarize. Very nice. So as the second section has depolarized, the first section will go back to its resting state by repolarizing, which means the inside once again becomes negative as you see me drawing it, and the outside becomes positive again. Okay? Do you see how this cartoon is getting very busy? And if you're not part of the conversation of understanding what everything means at different moments of time, it's going to be hard to understand this. Okay? So I'm hopeful that the analogy of the wave at a football stadium, where one section sits up, the next section sits up, the first section sits down. Then the third section sits up, stands up, the second section sits down. Fourth section stands up, third section sits down, so on and so forth, okay? So the third section is going to depolarize. I'll just put DP for depolarization. The second section will RP, will repolarize. Section 4 is going to depolarize. Section 3 will repolarize. Okay, and you have this wave that moves laterally along the length of the neuron. Okay. All right. So at this time, what questions do you have? Is this working for you? Are you getting a general idea how the membrane of the neuron at different artificial sections of the neuron change their charge at different moments of time? And they're constantly depolarizing and repolarizing in sequence like the wave at a football stadium. Questions, gang? Okay, Emily, can a trigger of a traumatic event activate neurons like that? Yeah, absolutely. In fact, Emily, for you to even appreciate that something is traumatic, you're going to trigger this event. Okay, for you to have a memory recall of a traumatic event, to remember that this happened and feel bad about it or anxious about it, it's going to involve a series of these events in various areas of your brain. Okay. Other questions? Okay. All right. I hope you're okay with this. All right. Are you sure there's nothing you want to ask at this time? Okay. Not seeing anything, so I hope you're okay. What we need to do now is show you how this idea changes. Now, I guess I better redraw it. How this idea changes when we have a myelinated neuron. Okay, so I'm going to insert a new slide here. And we will draw some basic things again. Okay. So now we're going to have a nerve impulse. Nerve impulse in a myelinated. If I could hold on a sec here. If I could spell, that would be good. In a myelinated neuron. Okay, that reminds me, let me go back here to our previous drawing. Do you appreciate it this time from this drawing we just did, that basically what we have here at each section of bleacher is a depolarization and repolarization event, which means we have action potential 1, followed by action potential 2, followed by action potential 3, followed by action potential 4, and so on and so forth. Where at each section of the plasma membrane of a neuron, you have an action potential. But if you chain them together as one action potential, and a second action potential, and a third action potential, then we have the entire wave of signal, which is the nerve impulse. It's the entire choo-choo train. Okay, let's continue with nerve impulse and a myelinated neuron. Okay, so we have our neuron here. We have the axon. Okay. We have the inside and we have the outside. No surprises here. And I will put, once again, I will put these artificial sections in the membrane. They are artificial. I mean, it's not like there's a dividing wall or anything like that in the real biology. Okay. All right. Now, where we do need to talk about something that is real anatomy is that since this is a myelinated neuron, what kind of cells should be present? Since this is myelinated, what kind of cells should be present? Go ahead and type that into the chat window. Okay, Nick, good. Okay, Schwann cells, absolutely. Okay, so remember from your textbook, let me give you a page here. Remember from page 117, figure 7.3, that you find myelin covering the axon with individual Schwann cells wrapped around the plasma membrane of that neuron. So what that means is, in different sections of this drawing, we need to indicate the presence of a Schwann cell. Okay, And so note that the Schwann cells don't literally cover every piece of the plasma membrane. They're sort of interleaved in between different sections. Okay, so right here in the drawing, these things are Schwann cells. These are Schwann cells, and the community or group of Schwann cells collectively form the myelin sheath. Okay, and notice that the areas of the neuron that are not actually covered with myelin, they're also given a special name. Can you tell me what the areas that are not wrapped in myelin are called? Can you tell me what the name of the exposed area of the neurons are called? Not dendrites, Quinn. Look very carefully at picture A of figure 7.3. Can anybody wrestle that out for us? Take a very good look at figure 7.3, picture A. What's the exposed area in between the Schwann cells called? Okay, Catherine, you got it there. Okay, little rough on the typing, but you have it. Nodes of Ron VA. Okay, so the exposed areas, let me type that into the chat window, are called nodes of Ron VA. There we go. All right, so these exposed areas have their own name as well. And let's indicate them in your drawing. Nodes of Ron VA. Nodes of Ron VA are named after Dr. Ron VA, who initially described them in the literature. Okay, so these exposed areas without Schwann cells are nodes of Ron VA. Okay. All right. So, long story short here, you're going to have the basic same sequence of events take place, which is to say, there will be some sort of stimulus which activates the neuron. Okay, now because this is a myelinated neuron, it's much more likely we're going to find this in an area of the peripheral nervous system because, you know, you might need an electrical signal to spread from the base of your spinal cord down to your big toe so you can manipulate your foot in some way, all right? And moving that signal down the length of your entire leg, which, you know, might be, I don't know, 25, 30 inches long is a long distance. So you're going to use myelin to accelerate the rate that the signal can be moved, okay? So I'm not going to redraw everything, but you have the visualization from last time. Here's what I need to tell you is different about a myelinated neuron, okay? For a myelinated neuron, you're going to have an action potential take place in the first section. So action potential 1 is going to happen. Because the next section of bleachers is wrapped in myelin sheath, it's wrapped by a Schwann cell, the second section of bleachers does not execute an action potential. Okay, there's no action potential. So the third section of the bleachers is the one that executes the second action potential. The next section of bleachers would be the fourth section, but it's wrapped in myelin sheath, so it's not going to execute an action potential. And so the fifth section of bleachers is the next exposed area that's going to generate an action potential. So if we think about our wave analogy, about what happens at a football game, think about a wave where you had the first section of bleachers stands up. The second set of bleachers has people that just don't want to cooperate. They don't want to participate in the wave, so they don't do anything. They don't stand up. But the third group of bleachers, they do want to participate, so they stand up. So in other words, the wave moves by having every other section of bleachers participate in the wave. In the case of the nerve impulse, when you have myelin, you have every other section of the neuron participate in the wave. Every other section of neuron that is not wrapped in myelin will stand up and sit down, will depolarize and repolarize. Okay, so we'll put down here, okay, in this scenario with myelin, the nerve impulse is able to cover the same distance faster because you actually execute fewer action potentials. Okay. The transmission speed increases because of fewer action potentials that occur over the same distance. Okay. Folks, are you okay right now? What questions do you have? Is this conversation making some logical sense to you? That when you talk about electrical signal, you literally are talking about the movement of, okay, maybe I need to ask you this. What is the electrical signal in the nervous system actually made of? What is the electrical signal? What is the electrical signal? What is it? What's it made of? Okay, Nick, you say potassium and sodium, and Quinn, movement of sodium and potassium. Okay, so are we moving atoms or are we moving charged atoms? Okay, Nick, we're moving charged atoms, so ions. So folks, the electrical signal is literally the motion of the ions back and forth across the lateral surface of the plasma membrane. That is the essence of the electrical signal, okay? When we talk about electricity, people think about the electricity running their lights at the house. And in that situation, the electricity is the movement of electrons between the atoms that make up the wires. In biology, it's a little different. We're not moving electrons, we're actually moving charged atoms. We're moving ions, okay? So the electrical signal is the motion of the ions back and forth across the plasma membrane, okay? Questions, gang? Is anybody passed out yet? Okay, some people are probably passed out. All right, very good. Let's continue. All right. Okay, so what we need to do next, folks, is hopefully take your maturing understanding of these processes and eventually wrap them back to the muscle physiology of chapter 6. Before we can do that, we need to talk about the next type of communication in the nervous system. Okay, so let's see where we are with with your notes here. Okay. All right. So in your notes, I have see PowerPoint drawing about electrical communication along unmyelinated and myelinated neurons. Okay. So hopefully the majority of that is making sense to you. Okay. What we're going to do next, well, let me put down number 4 here. And something I should have definitely done sooner is make sure you appreciate that the electrical signal is really referring to the motion of the ions. Okay. An electrical signal in this context is referring to the motion of ions, specifically sodium and potassium. I'll be honest with you folks, there's other ions involved. Calcium also plays a role, even though we didn't talk about any calcium here today. Okay. All right. So next, let's take the biochemical communication. And talk about that. Okay, biochemical communication involves the release of neurotransmitters. Okay. We're going to go over the prototypical example that we always give our beginning students, and that's fair enough. There's nothing bad about it. I mean, you got to start somewhere. But as you might imagine, just about every topic we talk about here does get considerably more complicated the deeper you get into things. Okay, a book like yours typically implies that most neurons of the body release a single neurotransmitter. That's not really true. Neurons can in fact release multiple different transmitters depending on a variety of circumstances. Okay, but what we're going to do is talk about the release of the neurotransmitter that you see on pages 120 and page 121. Okay. Okay. So to do that, I think I'm going to have to go back and do another horrible drawing for you. Okay. So let's get a new slide in here. And we're going to make this biochemical communication. Okay, involving neurotransmitters. Okay. So the basic way that this works, and that perhaps you have an idea of already from your reading, is that you're going to have a presynaptic neuron. Okay, so I'm gonna draw that kind of like this. Obviously, your book is a lot better at things than I am. Okay, so this would be the presynaptic neuron. And then we have typically a postsynaptic neuron, which I'll just draw a section of its plasma membrane here. Okay, this is going to be the postsynaptic neuron. Okay. There's some terminology that's going to come up here and that you definitely want to be familiar with. Okay. The area where the two neurons are connected to each other, they're not physically connected, but they're physically in proximity to each other. They're associated with each other. This area here is the synapse. Okay, so the synapse is the area where the two neurons are very close to each other. The space that's literally in between the two neurons has its own name. That's the synaptic cleft. So in other words, this space here is the synaptic cleft. All right. The synaptic cleft is filled with synaptic fluid. All right. So please don't think that this wide open space here is just a bunch of emptiness, like it's empty air or something. It's not empty air. Most spaces in the body are not open in an air capacity way, unlike the lungs, but they're actually filled up with liquid, right? So in this space, you're going to have synaptic fluid. Which of course is made of water and a bunch of dissolved solutes. Okay. All right, so what's going to happen here is that some kind of stimulus is going to activate the presynaptic neuron so that we have this chain of events that we spent, a half an hour describing already and drawing about is going to occur, where you're going to have a series of action potentials come down the presynaptic neuron. Okay, so I'll just indicate like this. AP1 goes to AP2, goes to AP3, right? This is going to spread down to the end of the neuron, okay? This end of the neuron here, right here, this bulb-like structure is called a synaptic knob. A synaptic knob. That's where the neurotransmitter is going to be released from. But before we can talk about that, let's see, I'm going to reserve this area below for notes. Okay, I'm going to reserve it for notes. Okay, so when the action potential, when this series of action potentials that collectively we call a nerve impulse reaches the end of the presynaptic neuron, when it reaches the synaptic knob, something's going to enter the presynaptic neuron. Okay, that may not be clear from the reading of your textbook. I'm not sure that your book goes into quite as much detail as I would want you to know. Okay, but okay, so let's see here. Letter A. Okay, so letter A down here in the notes are nerve impulse. Nerve impulse reaches synaptic knob. Okay, folks, when that happens, one of the things that I don't believe your book is telling you is that there are special channels, special proteins embedded in the plasma membrane of the synaptic knob, which are actually calcium channels. Okay, so I'm going to draw this thing here as a calcium channel. And this is one of the reasons I said a long time ago that having the right amount of calcium in your body, because you have the proper vitamin D to absorb it, is so very important. So what happens is the electrical signal at step A here reaches the end of the synaptic knob, so the calcium channel actually opens up. It opens up in such a way that calcium ions that are present in the synaptic fluid are able to enter the neuron. Okay, so here we have, I'm going to draw some dots here. These dots are meant to be calcium ions, Ca+2. So the nerve impulse reaches the synaptic knob, the calcium channel opens up, and calcium will literally enter through the calcium channel, it will actually enter the synaptic knob. Okay? So that would be note B here that we want to put down for you. Okay? Calcium channels open. And calcium, Ca+2, enters the synaptic knob. Okay. Folks, do any of you know why calcium has to enter the synaptic knob? What is the role of calcium here? Okay. What does calcium do once it enters the synaptic knob? Question in the chat window. What does calcium do once it enters the synaptic knob? Okay, and I admit this might be a little bit deeper than your book gives you. Do you have any idea, or can you think about this? What do you think calcium is going to do? Why do we need calcium to enter the presynaptic neuron? Remember, calcium entered the presynaptic neuron in response to the nerve impulse in the presynaptic cell. Well, think about it, gang. Calcium is often used by the body, used by cells to trigger events, right? In chapter 6, the presence of calcium allowed tropomyosin to roll off the surface of actin so that myosin could reach up and grab it, right? So calcium was pivotal in chapter 6. Well, here in chapter 7, calcium is also pivotal in a different way. Anybody want to hazard a guess? Okay, everybody seems a little tired here today. All right, so let me tell you what's going to happen here. The reason calcium has to enter the presynaptic cell is because calcium is the trigger that gets the presynaptic neuron to release the neurotransmitter. Okay, so before I draw that stuff in, can you tell me, question coming through the chat window, what structure inside neurons contains the neurotransmitter? What structure inside neurons contains the neurotransmitter? Okay, Catherine, not the synaptic cleft, no, but thank you for trying. It's not the synaptic cleft. Try again, Catherine, because you got one of the words right. Okay, Fiona, very nice. Okay, the synaptic vesicles. So you see from drawing that's better in your textbook on page 120, 121, okay, that in the presynaptic neuron, you're going to have little bubbles. They are lipid-based membrane bubbles, okay? And I'll just draw a couple here. These little bubbles are the synaptic vesicles. Okay. And these synaptic vesicles contain the neurotransmitter. Okay. NT for neurotransmitter. Okay. So when calcium enters the presynaptic neuron, it actually binds to molecules on the surface of the synaptic vesicles and triggers machinery inside the cell to pull that along the cytoskeleton till it gets to the border of the cell. So in other words, calcium triggers the motion of the synaptic vesicles so that the synaptic vesicles will migrate to the border of the plasma membrane and fuse with it. Okay, so letter C here. All right, letter C in our notes. Calcium ion, Ca+2, trigger the motion of synaptic vesicles. Okay, and what's going to happen is the synaptic vesicle will fuse with the plasma membrane of the neuron. The exterior surface of the plasma membrane is literally going to peel away. It's going to degrade. It's going to fall apart. And the neurotransmitter is going to be dumped into the synaptic cleft. Okay, now I'm just drawing one big NT symbol in your notes here for a neurotransmitter. But please understand that millions of neurotransmitter molecules could be spilled into the cleft here at this moment of time. Gang, do you have any questions right now? Okay, all right, so Quinn, do any neurotransmitters get lost? Invariably, yes, that probably does happen. Neurotransmitters, at some moments, neurotransmitters may fail to reach their intended destination. That's what I mean by lost, okay? It's not like it leaves the body or anything, but, you know, think about being along a riverbank. Okay, pick any river, Susquehanna River. Pick a river and imagine yourself on a riverbank, and imagine you throw a tennis ball into the river with the goal that it should migrate to the other side of the bank, the other side of the river. Well, maybe it never reaches that location because the current of the river is too strong for it to get across. In a very rough analogy, it is possible that some neurotransmitter will never do anything meaningful, okay? But, you know, fortunately for most of us, usually enough neurotransmitter does reach its target to do what we need to have done, okay? Other questions? Okay, let's keep rolling here. All right, so the neurotransmitter is now in the synaptic cleft, and it has to physically move over to the membrane of the postsynaptic neuron. So how's it going to get there? That's my question to you in the chat window. What method of transport does the neurotransmitter use to reach the postsynaptic cell? What method of transport does the neurotransmitter use to reach the postsynaptic cell? Emily, you say diffusion. Are you talking about simple diffusion or facilitated? Nick, you say active transport. Okay, so it won't be active transport, Nick, because the cargo is moving from a high concentration to a low concentration. So active is used to go the other way. So it's not active. Emily, is this simple diffusion or facilitated? Can anybody answer that question? Can anybody help Emily out? Okay, Quinn, you say facilitated. I can maybe understand why you would say it, but I'm talking about this cargo that I'm underlining on your screen. How does it get from this location to this location? It's going to be simple diffusion. It's going to be simple diffusion, okay? All right. So the neurotransmitter reaches the membrane of the postsynaptic neuron, and the neurotransmitter is going to bind to a neurotransmitter receptor embedded in that plasma membrane. So I'll just label this as an NTR. Okay, the neurotransmitter binds to the neurotransmitter receptor. And when that happens, that triggers the first event of an action potential called what? When the neurotransmitter binds to the neurotransmitter receptor, what's the first stage of an action potential called? Okay. Folks, I ask all the questions because I need to know that your brain is on and that you're thinking. Okay. When the neurotransmitter binds the neurotransmitter receptor, it will execute the first stage of an action potential, which is called what? Okay, Emily, good, depolarization. That's right. In other words, when the neurotransmitter binds to the postsynaptic neuron opens sodium channels so that sodium can enter, and that's called depolarization. So binding of the neurotransmitter, which was released by the presynaptic neuron, binding of that neurotransmitter cargo to the postsynaptic neuron will trigger an action potential, a nerve impulse in the postsynaptic neuron. Okay? And so I know that's a lot of words there. Let me simply put letter D in here, and let's put that down in your notes. Okay? The neurotransmitter binds to the postsynaptic neuron. Neurotransmitter binds the postsynaptic neuron, okay, which triggers depolarization in the postsynaptic neuron. Okay, once again, the neurotransmitter binds to the postsynaptic neuron, which triggers depolarization in the postsynaptic neuron. And so at this point, what's going to happen, I think you can maybe guess, that you're going to have a series of action potentials that are generated in the postsynaptic neuron, okay? And those series of action potentials as a collective train, like a choo-choo train, is going to be a nerve impulse. In other words, the nerve impulse in the presynaptic neuron triggers the release of a neurotransmitter, which will ultimately generate a second nerve impulse in the postsynaptic neuron. And this is how communication by the nervous system works in your body, how you have a nerve impulse in one cell, cause the release of a neurotransmitter, which then triggers a second cell to generate a second nerve impulse. And so it's kind of like hopscotch, right? You do one thing, then you release neurotransmitter, then you have a second action potential, on and on and on like this, over and over again, till the signal gets to its intended destination. Folks, do you have any questions right now? Okay. All right. So definitely, hopefully you've been able to take some good notes here, and I will send you this PowerPoint. But again, if you're not wrestling with the content, this picture may not mean too much to you, and you really got to think about it. Okay. So, so I will again reference, see PowerPoint drawing about neurotransmitter release. Okay. All right. So, because we're on the topic of neurotransmitters, I'm going to make a few comments about that. And then we're going to try to tie everything we've learned together between chapters 7 and 6 so that you can understand how the nervous system is capable of triggering muscle contraction in the body. Okay. Talking points about neurotransmitters and disease. Okay. There's as I said, there's probably about 150 to 200 different substances at this point that have been identified as neurotransmitters in the animal body. Undoubtedly, more of them will be discovered next year. Okay, so we're not going to try to explain all of these. That's more for a nervous system class or a neurology class, okay, to cover with you. But what I would like to talk is about a couple things that touch all of our lives here, okay? And so let me just mention a couple diseases where neuron behavior and neurotransmitter behavior seem to be abnormal, okay? The first would be Alzheimer's disease. Okay. Do you know anybody that has Alzheimer's? Are you aware of anybody? I mean, maybe somebody famous that has Alzheimer's. Okay, the person that comes to mind with Alzheimer's in terms of being famous is our former and now deceased President Ronald Reagan, right? When he passed, he had Alzheimer's disease. Okay, so you may know that patients with Alzheimer's, patients with this disease, okay, lose their memories, okay, so how horrible it would be to have a lifetime of memory with someone you love and care about, your spouse, and then you develop this horrible disease, and when your spouse walks in the room, you don't even recognize them anymore. Well, Peter, I can't answer your question because I actually didn't happen to watch Big Bang, so I don't know if Sheldon has that or not. I don't know, but so at any rate, so patients with this disease lose their memories, and it's an emotionally traumatizing disease. I said Alzheimer, not autism, right? The first one, Pete, the first one. Okay. So, no, obviously there are no cures for this disease. But what I can tell you is that for patients that have the Alzheimer's disease, what we do know is that there are neurons in the brain that are dying, okay, that are passing away, that are dying prematurely, okay? And because of this, they are not manufacturing the proper levels of acetylcholine in the body. Acetylcholine obviously being one of the major neurotransmitters of the body. Okay, so neurons in the brain that are dying prematurely, okay, acetylcholine in the body is abnormally low, okay? Now, we don't know, medical science at this point does not know why these neurons are dying. In other words, we don't know if the Alzheimer's disease is causing the death of the neurons or if the death of the neurons is creating the Alzheimer's disease. Once again, we have this chicken or egg scenario that I mentioned to you last time with multiple sclerosis. We really don't know the answer to this, okay? Okay, the scientific community cannot explain what is causing the neurons to die. Okay, we do not know the answer to that question. What is causing them to die? We don't know. But Unfortunately, folks, patients that have Alzheimer's disease have abnormally low levels of acetylcholine. Okay. All right. Number 2, Parkinson's disease. Okay. Okay, patients with Parkinson's, you may know, they have trouble coordinating muscle movement. You may know somebody with Parkinson's. The most famous person that I can think of, which you may know of, is Michael J. Fox, right? Michael J. Fox suffers with Parkinson's, and I believe maybe Muhammad Ali, before he passed away, may have been struggling with Parkinson's disease. Okay. Patients with Parkinson's have trouble coordinating muscle movement. Okay. Again, neurons in the brain that manufacture a different neurotransmitter called dopamine are dying prematurely. Okay. So the patients have abnormally low levels of dopamine neurotransmitter because of neuronal cell death. We also do not know why these cells are dying. We just don't know. Okay, it's an active area of research. Okay, and finally, a third disease where a neurotransmitter is implicated. So Alzheimer's, we have problems with unusually low levels of acetylcholine. Right? With Parkinson's disease, we have unusually low level of dopamine. Pete, is Parkinson's disease something to do with muscle response? Presumably so, yes, but exactly what's creating this disease is still a huge mystery to us. Okay. The third disease, which I think, fortunately for us, many of us will not suffer with Alzheimer's and Parkinson's, although some of us may as we get older. I mean, that's just reality. Okay. But there's a third disease that many of us will struggle with in our lives from a statistical point of view. We may have already struggled with it in our lives, and we may know people that actively struggle with it right now. And I'm talking about depression. Okay. Okay. Depression is not a momentary sadness. I mean, if someone in your family dies, you're going to be sad, right? That's not depression. That's sadness, and that's normal, emotional, healthy response. But depression is a prolonged, deep melancholy, a prolonged, deep sadness that you can't get rid of. Okay. I would say a multiple month kind of melancholy, a multiple month depression that you cannot shake. Characterized by a prolonged period of sadness. Now, there are many different subtypes of depression, and they're not all the same. That's one thing we're learning about. Not all depression is exactly the same. But one common form of depression that we know about is associated with abnormal production of the neurotransmitter serotonin. Okay. Many forms of depression are associated with abnormally low levels of serotonin neurotransmitter. Okay. And serotonin might be a molecule you've heard about because serotonin levels are also implicated in things like migraine headaches. Okay, so I hope you don't suffer from any migraines. But at any rate, for people that are depressed, they often have a correspondingly low level of serotonin neurotransmitter in their cerebral spinal fluid. Okay. And while we don't know exactly what's happening here, I do want to mention a common treatment for depression is to prescribe what are called SSRI medications. SSRI stands for selective serotonin reuptake inhibitors. Selective serotonin reuptake inhibitors, and their job is to prolong the amount of time that serotonin stays in the synaptic cleft, and therefore prolongs the amount of time serotonin has to activate the postsynaptic cell. These medications prolong the amount of time that serotonin stays in the synaptic cleft, increasing the chances that the serotonin will be successful in stimulating the postsynaptic cell. Okay. So going back to the bad drawing here, okay, what we did not talk about and what I'm alluding to in your notes at this time is that this neurotransmitter, right, this neurotransmitter, which I'll circle in green, right, this neurotransmitter, okay, it has a finite lifetime. It's only going to be in the synaptic cleft so long before either it is degraded and broken down by enzymes in the synaptic cleft that chew up neurotransmitter, or that the neurotransmitter in some cases actually goes back up into the original presynaptic cell to be reused again at a later time. Okay? But if that happens prematurely before the neurotransmitter can actually bind to the postsynaptic cell, then the postsynaptic cell is never stimulated. And we think, I mean, the scientific community believes that at least some aspect of depression is caused by postsynaptic neurons not becoming stimulated. So people stay in a depressed or melancholy state because the postsynaptic cells are not being activated. Okay. And so the SSRI medications that are out there, and there's various ones, I can't think of a name off the top of my head. There's a variety of them out there. They help the neurotransmitter remain in the synaptic cleft longer. Okay. All right, folks, do you have any questions at this time? Okay, everybody's quiet here today. So what we need to do to wrap all this content up, folks, is connect the dots. Okay, you think about it. I know some of you folks are a little bit young to remember 9/11 when we had the terrorist attacks in New York City and in Pennsylvania and Washington, D.C. But if you're old enough to remember the terrorist attacks, you know that one of the common themes in the news media and one of the common things that was talked about is that the citizens of the United States were complaining that our federal agencies of law enforcement missed the signs that there was going to be an attack. In other words, there was signs, there was data out there, but they did not connect the dots to prevent the attack. And folks, let's be honest, connecting the dots is probably the hardest part of learning. Memorizing something's not all that hard. It's laborious and tedious and not very fun, but memorizing what a synaptic vesicle is not that challenging. But understanding and connecting the dots of the physiology of how things actually work is extremely challenging. So what we're going to do next with our remaining time is connect the dots between nervous system behavior and muscle contraction. That's what we need to do, okay? So, do any of you have questions before we start that? Okay, I'm going to be challenging you with a whole bunch of questions as we go along here to see if you can connect the dots. Okay, so let's begin. Okay, I'm going to use PowerPoint to connect the dots. Okay. And so let's do this. Okay, let's connect the dots between chapters 6 and 7. All right, and so what we're going to be talking about is how the nervous system is capable of activating a muscle contraction process. Okay, so let me get the chat window up here again. Come on. Okay, chat window is back up. All right. So let's begin here, okay? What I need to draw is a neuromuscular junction, okay? So we're going to have the end of an axon, okay? So here's an axon, which you should now be familiar with from our conversation today, the end of it. And we also have, okay, what's going to be the plasma membrane of a muscle cell here. Okay, so we have the inside of the muscle cell, right? And this is the plasma membrane. Okay, and so this space here is the neuromuscular junction. Okay. All right. So I'm going to ask a series of questions. Okay. First question, what kind of thing, and I'm using the word thing as a very generic word here, what kind of thing will cause the neuron to depolarize? What kind of thing will cause the neuron to depolarize? Any questions? Okay, so good. I'm glad I'm getting the answers, folks. So look at everybody's answers. Matt, I see your question, and I'll address that later. So Nick, you say an action potential. No, Nick, an action potential will not cause the depolarization because an action potential is partly made of a depolarization. Okay. Sodium ions. Again, sodium ions are part of the chemistry of depolarization, but exactly. Okay, so Quinn and Matt both said stimulus or stimuli. That's what I'm getting at, right? There has to be a stimulus. So there has to be a stimulus so that you want to contract a muscle, right? Because right now we're talking about how the nervous system is going to contract muscle. So think about it. Maybe you're playing volleyball and you see the ball hit over the net, right? So your eyes literally see the ball. So light is bouncing off the ball and hitting the retina of your eyeballs, and it tells your brain, oh, there's an object there. And since I know I'm playing volleyball, I need to move my legs, to move my body over to the ball, to move my arms so that I can intercept the ball, right? So there has to be a whole bunch of complicated stuff that starts this chain of events, but it all starts with a stimulus. The stimulus in this case might be the reality that we're playing a volleyball game. You see the volleyball coming over the net, and you, in your mind, you think, well, I need to move my body so I can get to the ball. Okay, so there has to be a stimulus. Right, that stimulus is going to generate the nerve impulse, which we know is a series of action potentials. Action potential 1, action potential 2, action potential 3. Okay. All right. Okay. So let's say that the action potential reaches the end of this axon. What's the end of the axon called? What's the end of the axon called? Oh, I typed it to the wrong party here. The end of the axon. Okay, Emily, the space in between the cells is the synaptic cleft. But what's the end of the axon called? Okay, don't apologize, Emily. You're doing fine. Okay, it is, folks. It is okay to be wrong in class. I suppose it's not okay to be wrong on an exam, but it's okay to be wrong because we're learning here. Okay, so right, so the ax potential, the nerve impulse reaches the synaptic knob, and so if this is if this space here contains the synaptic fluid, right? In this area we have all this synaptic fluid. Can anybody tell me what substance is in this fluid right now that we need to enter the synaptic bulb? Okay, Nick, you say calcium. Fantastic. Awesome, 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 right? So the stimulus, whatever it might be the site of the volleyball, is activating a neuron, which is sending an action potential, a nerve impulse down, okay? And so when that nerve impulse reaches the end of the synaptic bulb, calcium, I'm really bad at drawing here, Ca+2, it's going to enter the synaptic bulb, okay? And when that calcium enters the synaptic bulb, what substance is going to be released into the synaptic fluid? What substance is going to be released into the synaptic fluid when calcium enters the synaptic bulb? The neurotransmitter. Okay, folks. Good. So Nick says neurotransmitter. Good, Quinn. The transmitters, right? The neurotransmitter is going to get released. So now I have maybe a harder question for you. And I will type it into the chat window. What is the name of the neurotransmitter released at the neuromuscular junction? There's a specific name. There's a specific neurotransmitter here. What is the name of the specific neurotransmitter that's released at the neuromuscular junction? Does anybody know from reading their textbook what it would be? There's all sorts of neurotransmitters in the body, but there's only one specific kind that we need to worry about for a neuromuscular junction. Pete, you win the cup of coffee. It's acetylcholine, okay? Acetylcholine. I will spell it out for you. It's got an L in there, okay? Acetylcholine. So calcium enters the synaptic bulb, and the neuron responds by releasing ACH for acetylcholine. Okay, so acetylcholine is ACH. And that's simply a molecule that would have been made inside the neuron. So acetylcholine, okay, we're talking in summary fashion right now, so that we can get through this. Okay, I do have my eyes on the clock here. We got about 12 minutes. So we saw a stimulus that activates the neuron, sends the nerve impulse down to the end of the neuron, the synaptic knob. Calcium channels open, calcium enters, triggers the synaptic vesicles to release acetylcholine into the synaptic fluid. Once acetylcholine is in the synaptic fluid, where does it want to go to? Where does it want to bind to? What does ACH, acetylcholine, want to bind to? What do you think, folks? Okay, Emily. It wants to bind to receptors on the postsynaptic cell, right? On the postsynaptic cell. Nicely done. These receptors would specifically be acetylcholine receptors. Okay, so embedded in the plasma membrane of the muscle cell are going to be acetylcholine receptors, ACHR, and the acetylcholine wants to bind to it. Okay, so acetylcholine binds to the acetylcholine receptors. By the way, next question, okay, because we always need to be thinking about what knowledge we have in the back of our head that we've already learned, and we need to connect the dots with new information in more advanced chapters. Okay, so another question: what category of organic macromolecule are acetylcholine receptors? What category of organic macromolecule are acetylcholine receptors? Thank you, Nick and Quinn. They are in fact proteins. That's right. Okay, and just because I've kind of forgotten, hint, hint, wink, 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 what are proteins made out of? Okay, Nick, amino acids. Nicely done. Okay, folks, if you want to connect the dots to a deeper and deeper understanding of any content, right, you got to remember what you learned chapters ago. Okay, so acetylcholine binds the acetylcholine receptors. That causes okay, now here's something that you may not be aware of. When you have an electrical signal spreading down the neuron, that's called a nerve impulse. Okay? A nerve impulse. It's a nerve impulse because it's happening in a neuron. When acetylcholine binds the acetylcholine receptor on the muscle cell, a new electrical signal will be created, but it's not going to be called a nerve impulse. It's going to be called a muscle impulse. A muscle impulse is basically the same thing as a nerve impulse, but because it's happening in a muscle cell, we call it a muscle impulse. Okay, so I now have action potentials spreading along the plasma membrane of the muscle cell, and if I'm honest, it's going to propagate in both directions. Okay, it's going to spread along the fabric of the plasma membrane of the muscle cell, and we will call this a muscle action potential or a muscle impulse. Okay. Now, in order to trigger the sliding filament model of muscle contraction, what substance must be released inside the muscle cell? What substance has to be released inside the muscle cell to trigger muscle contraction? Right? Connecting the dots, folks. What substance must be released inside the muscle cell to trigger contraction? So you have to get your old notes. Okay. Yes. Good, good, good. It's calcium, Quinn. Very nice. It's calcium, folks. Remember that. Calcium. Well, how in the world is the action potential along the surface of the muscle cell going to lead to a release of calcium inside the muscle cell? I'm looking at our time and thinking I would love to sit here and kind of watch you squirm as you try to figure that out, but I'm not sure we have time for that. Okay. Okay, so from looking at the picture in your textbook on page 106, please tell me what structure the muscle action potential travels down as it goes into the interior of the muscle cell. Do you know the name of this piece of anatomy? Okay, page 106. Page 106, figure 6.6, and the question is, what piece of that picture does the electrical signal travel into the muscle? Okay. Nick, I think you have it. Does everybody see what Nick is talking about? These tubules are called T tubules. T is short for transverse tubules. Okay, so on your drawing, what I want you to appreciate is that the electrical signal at the surface of the muscle actually goes into the interior of the muscle because the plasma membrane of the muscle actually has these invaginations, these internal extensions called T tubules, which are short for transverse tubules. Okay, transverse tubules are plasma membrane that go into the interior of the muscle cell. So, okay. So the action potential continues its journey down this way. Okay, I'm just drawing it as sort of an electrical lightning bolt at this point. So the electrical signal goes into these transverse tubules. Do you notice from the picture on page 106 that these tubules, which are yellowish gold in your textbook, seem to go in between some purple stuff? What's that purple stuff called? The sarcoplasmic reticulum. Thank you, Emily and Nick. The sarcoplasmic reticulum. Folks, in muscle cells, the sarcoplasmic reticulum is the muscle-specific version of the smooth endoplasmic reticulum. And the smooth endoplasmic reticulum of the muscle cell is a location that stores calcium for the muscle cell to use when it needs it. So inside the sarcoplasmic reticulum, which I will draw purple like your book, okay, and I'll put it around here. Inside the sarcoplasmic reticulum is calcium, okay? So when that electrical signal goes down through the T-tubule, the calcium inside the sarcoplasmic reticulum spills out. Okay, so the electrical signal at the surface of the muscle cell is able to trigger the release of calcium that's stored internally in the muscle cell itself. So let's think about this, and please give me another minute or two. I promise I won't take too long in lab today. But the stimulus, the volleyball, generated a nerve impulse in a neuron, which caused acetylcholine eventually to be released from the neuron, which then triggers a muscle action potential in the muscle cell, and the muscle electrical signal sends down the T-tubules and triggers the opening of channels in the sarcoplasmic reticulum to release calcium. Okay. All right. So walk me through it now. Okay. Walk me through it now. I'm going to stop sharing my screen. Walk me through it. The calcium has been released from the sarcoplasmic reticulum. Emily, what is the calcium going to bind to inside the muscle cell?
The myosin, or I'm sorry, can you hear me?
You're a little faint.
Okay, I apologize. I said the myosin, I can't actually remember. Sorry.
Okay. It's not the myosin, but you kind of have part of it. Quinn?
Isn't it like the topin, like type? Trying to
Okay.
pronounce it, trying to figure out how to pronounce it.
Yeah, it's troponin, right? Calcium binds to troponin,
Troponin.
which creates a domino effect of shape changes in the proteins, so that what protein shifts its position?
Oh.
Tropomyosin. Tropomyosin shifts its position so that what protein can form a cross bridge with what other protein? Okay. All right, I know everybody's getting tired and this is pretty heavy, but this is what connecting the dots is all about, folks. You need to not only know the pieces of the puzzle, you have to know how they relate to each other. You have to know how they connect together. Otherwise, we're not really understanding. So Quinn, you say, so the holes in actin can be shown so myosin can get in there to pull the actin. That's basically correct, right? Tropomyosin shifts away, myosin lifts up and binds to actin, forming a cross bridge. Myosin then ejects the AD and PI to form the power stroke, so we shorten the sarcomere. Then another molecule of ATP goes and binds to myosin, so it releases its grip on actin. Myosin acts as an enzyme and breaks down the ATP, so you can reposition the myosin head, so you can start another round of the sliding filament cycle that we went through from chapter 6. Okay, I think I've beat on you enough, okay? Folks, learning is hard work, okay? It really is. So this is what I expect of you during your study time, that you try to connect the dots. Okay. We're going to take a little rest here and come back for our lab time. Okay, let's start lab at 2:20, and we're just going to review the homework that was assigned to you. So I don't anticipate it will take more than 30 to 40 minutes. Okay, I don't anticipate more than a half an hour, maybe a little longer. Okay, how long it takes kind of depends on how well you completed your assignment. Okay. Okay. So let me see here. Is Matt on with us here? Matt, are you still with us? Okay, Matt's not with us. Okay, he's having a technical issue. Okay, folks, go take a break. Be back at 2:20. Okay, see you soon. Okay, gang, so what we'll do is go through the lab homework that was assigned to you, and I'll just go around the virtual room here, give you each a chance to contribute, and if there's any confusion or things need to be corrected, I'll help you out with that. You know, if we were in lab together, I would actually hand you, we have at school, we have these things called bone boxes that have entire skeletons in where the bones are separated, meaning they're disarticulated from each other. And you can kind of start putting the bones together like a giant three-dimensional jigsaw puzzle. So it's a lot, you know, it's a lot more interesting to do this sort of lab when you have things that you can put your hands on. Also, in the past few years, we've also gotten some very special software that we can use on tablets to help teach anatomy. Let me just, I'll pull it up here on my home computer and show you what it's like. Okay, so bear with me a second. Okay. All right, so for anybody that's interested in medicine, or if you're at all thinking about the possibility of continuing your education in a medical arena, you know, certainly learning anatomy is going to be a big part of that. It's always a big part for beginning students to get up to speed on anatomy. And one of the pieces of software that we are using with students these days is called Visible Body. Okay. So there's actually a company called Visible Body. You could Google it, and we're using the Atlas the virtual Atlas with our students at this point. But you know, just as an example, you can see on your screen that, you know, here's the entire skull. So wouldn't it, you know, we can use this with you at school. Unfortunately, I can't use it with you here when you're all at home, because you'd all have to purchase it for yourself. But at any rate, it makes learning the anatomy more enjoyable, makes it sometimes, you know, more complete and comprehensive, because you can do crazy things with this, such as, you know, if I want to look down into where the brain would be, I can select a bone and I can hide it, right? And I can look into the interior cavity of your cranium, so I could see structures inside that you wouldn't normally see. So it really does have excellent value for anatomy education. Okay, so all that kind of good stuff is there, you know, and you know, when you're learning the heart, things like that, we have all this kind of stuff available, right? The valves of the heart, the aorta, all kinds of stuff. And suffice it to say, unfortunately, you won't get a chance this summer to play with this because we're not together. Okay, so but if you're interested, just Google Visible Body. Okay, Visible Body, and you'll find the company that sells this software. Okay. Okay, gang, so let's take this page by page. Let me get my notes organized here. All right. So, okay. So I'm just going to use the screen here and use your name as I see it written on the screen. So, Catherine, would you be able to tell us the answers that you have for activity number 1 on page 40? Just go number by number and state what you have.
Okay. Frontal bone for number 1, occipital bone for 2, foramen magnum for 3. Number 4 is temporal bone. Number 5 is parietal bone. 6 is maxilla bone. And then 7 is mandible.
Okay, Catherine, thank you very much for your contributing. There were some errors there. Okay, so we need to fix that. Okay, here you go, folks. Number 1 is the parietal bone. Number 1 is parietal. Number 2 is occipital. Number 3 is the foramen magnum. And please understand, folks, the foramen magnum is not a bone. It's simply a hole. It's the hole that your spinal cord comes out of. Number 4 is the frontal bone. Number 5 is the temporal bone. Number 6 is the maxilla bone, maxilla. And number 7 is your mandible. Once more, number 1 is parietal, 2 occipital, 3 foramen magnum, 4 frontal, 5 temporal, 6 maxilla, 7 mandible. Do you have any questions about activity number 1? Okay, thank you, Catherine. Okay, so Catherine, okay, so you've done 1. So let's see, let's go to Quinn. Quinn, can you provide the answers for the skeleton on page 41?
Yep, I'm on it. Where's number 1? Number 1's the sternum. Number 2 is the invertible discs. I hope I'm pronouncing that right. Number 3 is the cervical vertebrae. Number 4 is the ribs. Number 5 is the thorax vertebrae. Number 6 is the lumbar vertebrae. Number 7 is the sacrum. And then number 8 is the how do I pronounce that?
Coccyx.
Coccyx.
Okay,
Yeah.
Thank you, Quinn. The answers that Quinn provided are correct. Let me just read them one more time.
Yeah,
Number 1 was sternum. 2 was the intervertebral discs. Intervertebral discs are not made of bone. They are made of fibrocartilage. Number 3 is the cervical vertebrae. 4 is ribs. 5 is the thoracic vertebrae. 6 is the lumbar vertebrae. 7 is the sacrum. 8 is the coccyx, right? Your tailbone. Okay, nicely done. Okay. Okay, Emily, can you please give us the answers that you put down for activity number 2 on page 42?
So for number 1, I had the pelvic bone. Number 2, I had pubic symphysis. 3, I had femur. 4, I had tibia. 5, fibula. 6, clavicle. 7, scapula. 8, humerus. 9, ulna. 6, radius. Oh, sorry, I think I just went out of order. I'll keep going though. 11, carpals. 12, metacarpals. 13, phalanges. 14, patellum. 15, metatarsals. 16, tarsals. And 17, phalanges.
Okay, thank you, Emily. There were 2 spots that you did have a bit of trouble with. You did switch radius and ulna. Number 9 is the radius.
Thank you.
Number 6 is the ulna. Also down towards the
Don't,
foot.
excuse me, don't you mean 10 is the ulna?
Yeah,
6 is
10.
clavicle.
Oh, I'm sorry. In my notes, number 10 looks like a 6. You are correct.
Oh,
Okay, so number 9 is radius, number 10 is the ulna. Down by the foot, number 15 is tarsals, 16 is metatarsals, 17 is phalanges. So let me read all of them again. Number 1, pelvic bone. 2 is the pubic symphysis. 3 is femur. 4 is tibia. 5 is fibula. 6 is clavicle. 7 scapula. 8 humerus. 9 radius. 10 ulna. 11 carpals. 12 metacarpals. 13 phalanges. 14 patella. 15 tarsals. 16 metatarsals, 17 phalanges. Okay, do you have any questions about that? Okay, if you got those or most of those, you're doing well. Okay, let's see. Pete, would you be comfortable reading your answers for page 43, activity number 3? Pete, are you okay to go ahead?
Yeah, I accidentally didn't fill them out, this part.
So Pete, you're very soft. We can barely hear you.
I didn't do this part because I forgot to do that part.
Okay.
Oh,
All
sorry.
right. Thank you very much for being honest about that. Okay, Jennifer, were you able to complete page 43?
I only have like half of those answers.
Okay. Fiona, were you able to complete page 43?
No, I wasn't able to get there.
Okay. Julia, were you able to complete page 43?
Yes.
Okay, Julia, please give us what you have. Start at number 1 and just go down.
1, mandible. 2, I don't know how to say it, but COCCYX.
Coccyx.
3, sternum. 4, ribs. 5, frontal bone. 6, lumbar vertebrae. 7, intervertebral discs. 8, temporal bones. 9, maxilla. 10, sacrum. 11, thoracic vertebrae. 12, foramen magnum. 13, cervical vertebrae. 14, parietal bones. 15, occipital bone.
Thank you, Julia. Were you able to do 16, 17, and 18?
I didn't.
Okay. All right, well, let's take the first 15. Folks, I have to say, there really is no reason you could not do this. Okay? So if we were in class together, I would be a little bit irritated. There's no reason you can't do it. So why wasn't it done? If you did it, you would know that Julia made 2 mistakes with her answers. So if you wrote down what Julia told you, there were 2 mistakes. So I will correct them at this time, okay? You need to participate in the course or you get nothing out of it. That is your responsibility. Number 1 is mandible. 2 is coccyx. 3 is sternum. 4 is ribs. 5 is frontal. 6 is lumbar. 7 is intervertebral discs. Number 8 is an area that Julia had wrong. Number 8 is occipital. OCCIPITAL, occipital. Number 9 is maxilla. 10 is sacrum. 11 is thoracic. 12 is the foramen magnum. 13 is cervical vertebrae. 14 is parietal. 15 is temporal. Okay. Now, 16, 17, and 18. We're going to sit here till you get it done. So I want everyone to figure them out and type their answers into the chat window. You have time. Go ahead. 16, 17, and 18. You do them. You may need to consult your textbook. Okay, folks. a lot of answers out, and thank you for doing that. If you compared answers between each other, you will notice that you did not always agree. So we need to clarify some of this, okay? All right. So remember, when homework is assigned, I expect you to make reasonable efforts to get it done. Number 16. Number 16 has 6 different bones that would be listed. 16 is the frontal bone. There are 2 parietal bones, 2 temporal bones, 1 occipital bone, 1 sphenoid bone, that's S-P-H-E-N-O-I-D, and 1 ethmoid bone. So again, frontal bone, parietal bone, temporal bone, occipital bone, sphenoid bone, ethmoid bone. Okay? Do you have any questions about number 16? Okay, so those make up your cranium. Number 17.
You said there were, oh.
Do you have a question, Quinn?
Yeah, you said like there were 2 parietal bones?
Yeah, on
Like,
both sides of the skull.
Oh, I thought they were just one big one.
No, your left and right side of the skull.
Oh, all right.
Okay.
I was just curious.
That's why we have you do this, right? So that you realize these things. Okay. Number 17, list the bones of the vertebral column in order from the neck to your tailbone. They would be your cervical vertebrae, your thoracic vertebrae, your lumbar vertebrae, your sacrum, and your coccyx. There should be no listing of cartilage there because question 17 asks for bones, and the intervertebral discs are not bones. Okay. Number 18. So Pete, 5 bones for 17. Not 5 bones, not literally 5 bones, Pete, but 5 groups of bones. Okay, 5 groups of bones. Okay, for number 18, the rib cage is made of your ribs, your sternum, which is your breastbone, and your thoracic vertebrae. That's it. Ribs, sternum, thoracic vertebrae. Okay? Is there anything on page 43 you need me to repeat or you need help with? Okay. All right, folks, let's continue to the next page, page 44. Is there anyone out in the virtual world there that would feel comfortable giving these answers?
I can do it since I didn't do the last one.
Okay, go ahead.
I'll try. I don't know if they're all right though. 1 is clavicle, 2 scapula, 3 phalanges. I don't know how to say that, sorry. 4 patella, 5, oops, I have a double phalanges. 6 ulna, 7 femur, 8 scalpa, 9 radius, I think. Yes. 10, I have tarsus, 11 carpalis, 12 metrocels. Sorry, I don't know how to say any of these. 13 sacrum, 14 pubic bone, 15 tibia, 16 fibula, 17 I have femur, tibia, patella, distal femur. 18 I have humerus, radius, and ulna. And then 19 I have scapula and clavicle.
Okay. Thank you, Jennifer. Thank you for contributing. There was a few mistakes in there, okay? So if you had different answers, you may not be completely crazy. So let's go over these again. Number 1 would be your clavicle. 2 is something that Jennifer misidentified, I believe. 2 is the humerus. 2 is the humerus. 3 is your phalanges. That's a P-H-A-L-A-N-G-E-S, phalanges. 4, your kneecap is your patella. 5 is the radius. 6 is ulna. 7 is femur. That's F-E-M-U-R. 8 is scapula. 9 is carpals. 10 is tarsal. So again, 9 is carpals with a C, and 10 is tarsals with a T. 11 is metacarpals. 12 is metatarsals. 13 is pelvic bones. 14 is the pubic symphysis, which is a piece of cartilage. 16 is the tibia. 15 is the tibia, sorry, 15 is the tibia. 16 is fibula. Number 17 is the femur, patella, tibia, and fibula. Number 18 is the humerus, the radius, and the ulna. And 19, the bones of the pectoral girdle. Well, the pectoral girdle is both on the left and right side of the body. So you have 2 clavicles and 2 scapula. Folks, do you need any of that repeated or clarified for page 44?
Can you repeat number 13?
13 is the pelvic bones. Okay, using the vocab language that is at the top of the page, it says pelvic bones. 13 is pelvic bones. Okay, anything else? Okay, Pete, you want, okay, 7 is femur. 17 is femur, patella, tibia, and fibula. 18 is the humerus, radius, and ulna. 19 is the clavicle and scapula. You have 2 of each of those on the left and right sides of the body. Okay, all right, anything else? Okay, folks, let's go to the skeletal muscles on page 45. Alyssa, would you be able to help us with that?
Yeah, I can. I have 1 as pectoral major, 2 as biceps bronchi. I have 3 as rectus abdominis, 4 as external oblique, 5 as the quadriceps femoris group, 6 as the tibialis anterior. Where did 7 go? Oh, 7 I think is the trapezoidus, the over shoulder blade, but it was that one I'm not sure about. 8 I have as the deltoid, 9 as the triceps bronchoid, 10 as the latissimus dorsus, 11 as the gluteus maximus, 12 as the hamstring group, and 13 as the gastrorhodomyces. I don't think I said a lot of those right. I'm sorry.
Okay, thank you, Alyssa. I really appreciate that. You did get all the answers correct, but you know, a few issues with pronunciation, and that's normal. So let me just say them one time for everybody. okay, but you did a good job. Number 1 is the pectoralis major. 2 is biceps brachii. Alyssa, you kept wanting to say bronchi as though we're talking about the bronchioles in your chest cavity. That's not the same thing. So 2 is biceps brachii. 3 is the rectus abdominis. 4, the external oblique. 5, the quadriceps femoris. 6, the tibialis anterior. Number 7 was the trapezius. 8, the deltoid. 9 is the triceps brachii. 10 is the latissimus dorsi. 11, the gluteus maximus. Okay, it may be of interest to you to know that your butt has the gluteus minimus, gluteus intermedius, and gluteus maximus. There's actually 3 muscles there that overlap on top of each other, but typically we can only externally see the gluteus maximus. Number 12 is the hamstrings, and 13 is the gastrocnemius. Okay, would you like any of that repeated or clarified? Okay, let's continue here.
I was a little curious about number 7. In the book, it just looks like it's like in a different place. Is there a reason that it's pointed to in different kind of ways?
Yes. Okay, Alyssa, thank you for asking that. That's because the trapezius is a very large muscle, so the line's only pointing to a section of the muscle. Okay, so let me see if I can bring it up here on my software. That's right, you're not seeing it right now, are you? Okay, hold on. Okay, so on the software, let me see if I can bring this up here. So that's the entire trapezius muscle on the right side and the entire trapezius muscle on the left side. Okay? That's why, folks, you know, learning anatomy is not a trivial thing, and learning it from little charts and diagrams in the book is really not the best way to do it. Okay? Okay. Does that make sense about the trapezius? Was that Alyssa that asked that? Okay, okay, all right. Anything else you need clarity on? All right. I mean, I do understand that many of you do not have an interest in a science or medical career. Maybe some of you do, but you appreciate that if you're going to be involved with medical care of any kind, you really can't make mistakes, right? Because then people get hurt or die, okay? And then there's big problems, and you got medical malpractice. Okay, so once you get into the higher level courses, the standards will be a lot more stringent. Okay, let's continue here. Okay, so on page 46, we have number 1 there at the bottom for flexion and extension, okay? Just number 1. Paquasha, would you be able to do the answers for question number 1 on page 66 and at the top of page 67?
I think it's extension.
Okay, the first question was flexion. That's correct. What's the next one?
Oh, the second, what is the name of the upper arm muscle?
Yes.
That one was flexon.
No, that's not the name of the muscle. Okay. Would anybody be able to do the questions for number 1? Okay. All right, let me help you with this. On page 66 at number 1, it says sit with your elbow resting on the lab table, bend at the elbow to move your hand towards your shoulder, right? So you're doing this, right? When you do that, that is an example of flexion. The next question, what is the name of the upper arm muscle which is contracted? That would be the biceps brachii. What is the name of the antagonistic muscle which is relaxed or stretched? That would be on the back of your upper arm, the triceps brachii. Triceps brachii. Okay.
No question.
Go ahead.
For number 1, I thought it was more of an extension, not a flexion.
So Pete, if I do this, if I start like this and I go like that, that's flexion. The reason it's flexion is because this angle is decreasing. This angle is decreasing as you go like this angle is getting smaller. That's why it's flexion.
I thought flexion mean like flexible.
Oh, you thought about flexible? Like you could stretch, you
Yeah.
mean? Yeah, you're confusing terminology there.
Okay.
Okay, so be careful, but thank you for sharing that because other students will likely stumble upon similar things and it's okay. But no, that's not what we mean. Okay,
We should like change the vocab for something else, no?
okay. So on page 66, if you look at where the word flexion is defined, it says decreasing the angle of a joint. So that's why this first one is flexion. Okay, so I can't really show you good through the internet, right? But if I were to measure the angle between this area of my arm, Pete, are you able to see? If you were able to look at the angle between here and here, that's 180 degrees. It's a straight line. And then if I bend, now it's more of a 90-degree angle, right? So I'm decreasing the angle, that's why it's flexion. Quinn, do you want to do the next ones? Are you asking
Yep.
to do? Okay.
I think I'd be able to do this efficiently.
So at the top of page 67, can you do the next?
Do you mean 47?
I'm sorry, I got two different versions of my book here.
I'm just like,
Page
67
47.
is alarm.
Can you
And
answer
then
the
I
next?
think it's, oh.
Can you answer the next 5 questions?
Oh, yeah, I can do that. It's extension because you're increasing the angle. If flexion's decreasing, then extension would be increasing the angle. So if you're doing this, well, I'm kind of limited since I'm sitting in my recliner chair outside, but yeah, you're making the angle bigger, so it's extension. The name of the upper arm muscle that will be contracted will be the tricep. The tricep brachii is the one that's going to be, oh, please don't do my paper. Will be the tricep brachii will be flex, and then the agonistic muscle will be the bicep brachii. And during push-ups, you're pushing yourself up. That means the tricep brachii will be working itself out. And then if you're doing dumbbell curls, which everybody knows those, every stereotypical gym rat does them, so
Right.
dumbbell curls are using your biceps.
Right. Arnold Schwarzenegger arms, right?
Yes.
Okay. Good job, Queen. She's right. So let me just read that off again. The top 5 questions at the top of page 47 are extension, triceps brachii, biceps brachii, triceps, and biceps. Okay. Now, number 2, folks, I'm sorry, I can't really demonstrate that for you, at a distance here because of the way the camera is. But does anybody can anybody take a crack at the questions under number 2? Anybody want to try that?
I can do number 2.
Okay.
I have extended for the first answer, which I think is right.
Yes, it is.
And then the second answer is the one that I really have trouble saying, but I believe that it's the gastroenemesis.
Gastrocnemius, yes.
Gastrocnemius, sorry. Number 3 is the tibulus anterior. And number 4 would be flexed, and then it's the opposite. So the tibulus anterior, and then, oh my gosh, I forgot how to say it again.
Okay, Alyssa, thank you. You have that correct. Let me read it again for those that maybe had trouble hearing. Number 2, the first question is extended. The second question within 2 is gastrocnemius. The third is tibialis anterior. The fourth one is flexed. The fifth one, tibialis anterior. The sixth one is gastrocnemius. Of course, the hope is at home that you would actually do these activities and pay attention to how your own body feels, right? When you stand on your tiptoes, what muscles feel tense? What muscles feel relaxed? Okay? All right. Number 3, okay? Being conscious of our time, let me start handing more of the answers out for you because I do want to give you some available time for your lab exam that still has to be done. Okay, number 3, it asks you to go through the motions if you were to kick a ball. I'm sure you've all kicked a ball at some moment in your life. The first question, does this action result in your lower leg being flexed or extended at the knee? It would be flexed, right? Flexed. Next question, what group of thigh muscles is responsible? This would be your hamstrings. When you move your lower leg forward to kick, does this action result in your lower leg being flexed or extended? That would be extended. What group of thigh muscles? That would be the quadriceps femoris muscle group. Okay. Do you need any of that repeated? Okay. Spend time doing the work, feeling the muscles in your legs moving. Okay. That's what we want you to do. Okay, number 4, demonstrate a movement that would be an example of abduction and adduction. Well, okay. Did any of you ever make snow angels growing up as a child, where you would lay down in the snow and flap your arms and legs to make a snow angel imprint in the snow? So as you swing your arms away from your body to make part of the wings, that would be abduction, ABD. So this motion is abduction, and bringing your arms back towards your body would be adduction, ADD, ADD. Okay? All right. Number 5, demonstrate a movement that would be an example of rotation. Well, folks, we don't have to make this hard. Rotate your head left and right. Okay, there you go. That's rotation. Okay, so if you're looking to the left to talk to your family member, that's rotation. Demonstrate a movement that would be an example of circumduction. Okay, well, there's things you could do here. It's kind of hard to demonstrate, but okay, if you can see me at all through the video camera, if I take my arm and I rotate it like this, that would be an example of circumduction. If you can envision a geometric object being formed through the motion of my arm from my shoulder to my fingertips, what geometric object would I be outlining with the motion of my arm? What geometric object? Okay.
Like a circle.
Okay. So Emily, the tips of my finger form a circle. But what about the rest of my arm? Can you envision Yes, Alyssa. It would be a cone. It would be a cone with the point of the cone here at the shoulder. It would be a cone, okay? See if you can prove that to yourself at home. You know, people get embarrassed. They don't want to do this stuff in front of other people, but folks, you got to do these things if you want to understand. Okay, number 6, rotate your lower arm from palm forward to palm rearward, okay? That's really, really hard to demonstrate. I don't think I can. You know, it's like this, and I go like that. So from here to here, that's pronation. Here to here is pronation, and the opposite of that is supination. Okay? The second part of that question, how does the positional relationship of the radius and ulna change in pronation compared to supination? Well, basically the bones of your lower arm, the radius and ulna, as you move, they're going to rotate across each other. Okay, they're going to cross over one another. It's kind of hard to appreciate what from a tiny little window screen here in front of you. Okay. Do you need any of numbers 4, 5, or 6 repeated? Okay, then activity number 7. Let's close this up. Number 1 is the trapezius. 2 is the rectus abdominis. 3 is deltoid. 4 is pectoralis major. 5 is latissimus dorsi. 6 is external oblique. 7 is the gluteus maximus, the gluteus maximus. Folks, do you need any of activity 6 repeated? Okay. All right. Then we've come to the end. Folks, there are going to be some activities in the weeks ahead that I ask you to do at home. Please don't make it so that I have to require you to hand in work. Okay? Make sure that that work is getting done as it should be getting done. Okay? All right. Do you have any questions before we leave for today? And obviously you need to finish your lab exam. Okay. Folks, thanks so much for your time today. Have a nice weekend. Pete, go ahead.
I didn't see the microphone. What kind of activity do we have to do before the next lab?
I will tell you more about that on Tuesday, but if you want to be looking ahead, there are some things in lab number 5 that you can kind of do on your own at home that I will be giving you some notes about, okay? For example, just as an example, Pete, if you want to start looking at things, on page 53 of your lab manual, activity number 2, the 2-point touch discrimination test. All you need to do that is a paper clip and another human being, so a friend or somebody you live with, and you can actually do it, okay? So I'm not assigning it yet, but I will be assigning it on Tuesday, and if you want to start looking ahead, that might be a good thing to do. Okay, Jennifer, you want me to repeat number 6. Okay, hold on a second. So going back to activity 6 from our current lab activity, our current lab exercise. Jennifer, number 1 is trapezius. 2 is rectus abdominis.
Sorry to interrupt you. I meant to say the pronation and spination. That one.
Oh, okay, thank you.
Sorry.
Okay. For number 6, the first question is this movement pronation or supination? It would be pronation. So you should do it at home and understand that that motion is pronation. And then the second question, how does the relationship of the radius and ulna change? Well, in simple terms, they cross over each other. They cross over each other. Okay? All right, so folks, be looking forward. If there are parts of Lab 5 that you can reasonably do, such as have a paper clip, have a friend help you do something, I'm going to be assigning that, okay? Because that's not a burden. I think you can find a paper clip and a friend, okay? Do you have any other questions? Okay. Have a great weekend, stay safe, and don't forget to take your lab exam, okay? I'll see you next week.